Hey guys, Mr. Gibson here. So for today, you guys are doing the explain activities for the DNA to protein section of this unit. What this means is we are nearing the end of Unit 1. So you will have a test coming up before too terribly long. So make sure you get the full use out of these activities. So up to now, you guys have done this. You've done these 3 explore activities. One was 2 parts. Hopefully that helped a lot. This gizmo everybody loves. In the past, everyone says it really helps them understand how proteins get made. So hopefully that helps you guys out. Hopefully you have put this vocabulary into your interactive notebook. And now you are on the explain 1 and 2 activities, which are both due today. So what you guys are going to be doing for those is we have a Stemscopedia again. Again, that looks like this. Okay, there's your Stemscopedia. And for the first assignment, it's going to be graded in your notebook. And this is a new literacy strategy. So what you guys, when this loads, there we go. Just a second. All right, there we go. So here you have a vocabulary table. So what you're going to do is this. Before you read anything, you're going to go through and look at these words, these here and these here. And if you, so like genome, if you have no idea what that means, you just type in no clue, okay? If you see DNA, hopefully you know what DNA is, but if not, whatever. You'll learn today. So if you know what DNA is, you put know it. I am good to go on DNA. If you see chromosomes and you're like, I've seen it, I've seen that word used, but I'm not solid on what that means, you're going to type in seen it. And that's purely for you. There is not a right or wrong answer here. It is just your opinion, whether you have no clue, if you remember seeing it, but you're not sure what it is, or if you for sure know it. And then what you are going to do is, as you read, you're going to look for these words, okay, and fill in your own version of the definition here. Now, for some of these words, their definition might be really long. So what I want you to do is just paraphrase it in your own words, what these mean. You're going to find them in the Stemscopedia, and I want you to paraphrase it here. And what that's going to do is really help take what those words mean and deposit that information into your long-term memory. Now, really quickly, for those of you who really like things explained to you, I want to go through and just generally explain the main points of this Stemscopedia, okay? So this whole second part of Unit 1 is all about how DNA is used to make proteins, how we get from DNA to proteins. So what's really important is to understand that DNA carries protein instructions. I always like to use this analogy. Think of your DNA like a cookbook. Think of the proteins as the food you make using the cookbook. So the instructions on how to make all the food is found in the cookbook. And the food that you make using that instructions is actually what you're using, what you're eating. And so DNA has all the instructions in it for how to build all the proteins that your body needs, okay? And proteins do everything. Think it if anyone ever asks you what proteins are used for, they're used for everything, okay? Now our DNA is tightly wound into these structures called chromosomes, okay? Chromosomes, think of that as like a spool of yarn, except instead of yarn, it's your DNA. Your DNA is tightly coiled around and bundled up, and that just makes it really easy to move around in the cell where it needs to go, in the nucleus, okay? So if we're using that analogy that the DNA is like a cookbook, the proteins are the food, the individual recipes are the genes. Okay, genes are just sections of DNA, and specifically they are sections of DNA that code or hold the instructions for how to make a specific protein. So for every protein in your body, there is a gene in your DNA that has the instructions for how to make that protein. So like your hemoglobin in your red blood cells, there is a gene that has the instructions for how your body's supposed to make hemoglobin, okay? Kinesin, pepsin, all of those are, all the enzymes, everything you can think of that's a protein, lactase, amylase, all those different proteins all have genes that have the instructions for how to make that protein. So DNA is the whole cookbook. Each individual recipe would be a gene, and the instructions in the genes are used to make specific proteins. Now, there are steps by which this must happen, because here's the thing, your protein factories, the organelles in your cells that make proteins, are called ribosomes. Those are in the cytoplasm of your cell, which is outside of your nucleus. All of your genes, all of your DNA is inside of the nucleus, and it can't leave. Your DNA can't leave the nucleus. So how in the world, if all the instructions are in the nucleus, the things that need the instructions to make the proteins are out of the nucleus, how do the instructions get from the DNA to the ribosome so that you can make proteins? Well, two main steps have to happen. And those two steps are transcription here and translation. Okay, so what happens is your DNA, let's say we need to make hemoglobin. Let's just use that as an example. Okay, let's say your body needs to make hemoglobin. There's a specific gene in your DNA that has the instructions for how to make it. Okay, so what happens is an enzyme will go in and copy down those instructions. It will copy down that gene, the instructions in that gene for how to make hemoglobin, and it will copy it into a strand of RNA, which, if you look over here at this picture, is slightly different from DNA. RNA is single-stranded. It's not double-stranded like DNA. It's relatively short because it's only copying that one gene. And you guys know about A, T, C, and G, those 4 nitrogen bases that are in DNA. RNA is a little bit different. It has A, C, and G, adenine, cytosine, and guanine, but it doesn't have thymine. It doesn't have the T. Instead, it has this nitrogen base called uracil. Okay, so there's a difference. And there's a difference in the sugar that makes up its backbone. It has ribose instead of deoxyribose. But the main thing here is that RNA, its job in this case is to copy down the instructions for that one gene. Because RNA, it's so small, it can leave the nucleus. So RNA goes in, copies down the instructions from that gene for how to make hemoglobin, just to use our example, and then it leaves the nucleus. And it carries that to the ribosome. And that process of copying down the instructions is called transcription. If I were to ask you to transcribe something, that literally just means you would copy it down word for word. If I handed you a note and said, hey, transcribe this for me, you would just copy it down. Okay, doctors have to transcribe notes. There are a couple of different jobs where people have to transcribe information. They're literally just copying it down. And that's what our RNA is doing. It's copying down the instructions for how to make a protein and then taking those instructions out of the nucleus. Now, once that happens, okay, here. Okay, so once that happens, the ribosome, which is your protein factory, will take that RNA, which in this case specifically is mRNA. That M stands for messenger. So this is an RNA strand that's carrying a message. Okay, it's carrying the message from the DNA, so we call it messenger RNA. The ribosome will just read that. It will feed into the ribosome and literally just read the instructions. It will read the codons or the sequences of letters that tells it, okay, I need to make this specific protein. As it reads it, this process of translation takes place. Okay, so transcription is copying DNA into RNA. Translation is where the ribosome reads the RNA, uses the instructions to make the protein. Specifically, here's what happens. Okay, proteins are made of these things called amino acids, and you see some of them here. They're not nice acids, they're amino acids. Hopefully you're laughing at that on the other side of the computer. Amino acids are a totally different ballgame from DNA and RNA. These are totally different structures, and later on we're going to get into what amino acids actually look like. But right now, all you need to know is that they are the building blocks of proteins. So when we add multiple amino acids together, we have a protein. So what the ribosome does is it reads the mRNA, and it has all the instructions for what amino acids it needs to build the protein and what order to put them in. And so as it reads it, the ribosome doesn't have the amino acids right there to do it. These other structures are floating around in your cytoplasm in the cells, carrying the amino acids around basically on their backs. And those are called tRNA or transfer RNA. And we call them that because they transfer the amino acids to the ribosome. So what happens is the ribosome will read a sequence of letters on the mRNA. For example, let's say AUG, okay? Adenine, uracil, guanine. If it reads that sequence, that one is what's called a start codon, but it also is, it it's the instructions for the amino acid methionine. Okay, you don't have to memorize that, but I'm just using this as an example. So when it reads AUG, it will know, okay, the first amino acid to make this protein is methionine. And so it will call up the tRNA carrying around methionine. That tRNA will bring it into the ribosome and drop it off. And it will do this sequence over and over and over and over, building one amino acid at a time, just like building a sandwich or something like that, until it reaches a signal that tells it to stop, like a period at the end of a sentence. Okay? Once it stops, those amino acids, that amino acid chain, like this one you see in the picture here, will break off from the ribosome and fold and become the functioning protein. So that's how we've gone from DNA to mRNA to proteins, okay? Now those proteins are super important because those are what tell the cell its function. Those are like the workers in a factory, okay? So, or the waiters at a restaurant, or the employees at any business. They are what sets cells apart from other cells. So this is what allows for specialized cells, because each different cell type has different proteins that are being made to give that cell its specific function. Okay, so like here they start as stem cells, and then the proteins in these are made to make them muscle cells. The proteins here, like hemoglobin, are made to make these red blood cells, and then over here you have like a neuron where proteins are being made to give it its function. Proteins, this is going a little bit deeper here, but some proteins are called enzymes. And enzymes are proteins that help our bodies with chemical reactions. Another word for those is catalysts, and a catalyst is basically just something that speeds up a chemical reaction. Okay, so whatever it's working on is called a substrate, and whatever comes out of it is a product. So some proteins are also making things. So the things that it's working on are the substrate, and the finished product is called the product. That's pretty simple, right? All right, so hopefully that made sense. If not, there are a ton of good videos that explain protein synthesis on YouTube, or you could always email me or call me for help if you need any further explanation. And this does a pretty good job of explaining it too. All right, guys, if you need help, let me know. Hopefully this is helpful. Good luck.